Yeah. I'm Hesna. Hello, everyone. I'm going to present 19F labeling of RNAs as a tool to analyze RNA-protein interactions in physiological conditions. So, before going to the details, my project is about alternative splicing, and I would like to introduce first what is alternative splicing. Alternative splicing is the process by which from one gene you can obtain different transcripts, so different isoforms of protein and different effects. For example, here I took the example of survival motor neuron, SMN. We are in the nucleus, so from the DNA, after transcription, you will have this pre-mRNA, including exons and intros. Then, during the alternative splicing, depending on which protein will intervene, RBP1 or RBP2, RBP is for RNA binding proteins, you will have either this transcript, mRNA1, with the removal of only the introns, and you get exons 6, 7, and 8, and a normal physiological protein with a normal effect. If, in the other case, if RBP2 intervenes, in case of mutation, for example, you will end up with removal of introns, but also removal of one of the exons. So this is what we call the exon exclusion, exon 7 exclusion. End up with this transcript and with a truncated protein, SMN delta 7 unstable protein, and this leads to muscular spinal muscular atrophy, which is a lethal disease in infants. And that's why it's very important to understand the mechanism where alternative splicing is involved in, to explain when and how this protein will intervene for the splicing of the mRNA in case of the conception of drugs against this kind of diseases. So the aim of this project is to explain how do RNA proteins, RBPs, control the selection of sites for splicing. Do they compete? Because we know that, for example, in the case of SMN2, we can have up to 40 splicing factors intervening in this splicing. For the space given here, they cannot all fit at the same time. So we want to understand how they compete to bind to the RNA. Do they bind concurrently to the RNA? And if so, if there is any cooperation between them upon binding to the RNA. So we have 5 labs on this project, 3 in Leicester, the lab of Andrew Hudson, Ian Apron, and Cyril Dominguez, and 2 in Glasgow, Glenn Burley for chemistry and Alice McClare for engineering. Each lab has its technique, and in the Cyril lab, we work mostly by NMR to analyze the interaction between different splicing factors or binding domain of these splicing factors and a portion of the pre-mRNA, which I show here. So these are results published in 2014 about a portion of SMN2 pre-mRNA here, and the different potential sites of different splicing factors intervening in the splicing of SMN2. And to work by NMR, we decided to start with a small oligomer of 21 bases and to use the NMR of the fluorine to analyze the interaction of this RNA with 3 different splicing factors, A1, SISF1, and some 16s, because we already know the biology, the biochemistry of those proteins. To do so, we decided to label the RNA in different positions using fluorine. Why using fluorine instead of 13C or 15N or proton? This is what people usually use because fluorine has a high NMR sensitivity. It covers a wider range of chemical shifts than proton. The chemical environment strongly affects its chemical shifts, and it can be incorporated in many positions on the base and also on the sugar. And the good news is that 2'F nucleosides, which is fluorinated nucleotides on the sugar, are commercially available. The fluorine has already been used to prove RNA-protein interactions before, but never has been used to study single-stranded RNAs identify protein binding sites on one RNA, and investigate concurrent or competitive binding of pairs of proteins on one RNA. And here is the novelty of our work. So this is our strategy. What we do is we take this portion of RNA that we bind directly with water, fluorinated into positions, and we analyze its interaction with the binding domain of three proteins, SRSF1, A1, and SAM68, that we overexpress in our lab and purify. There are two things important to know. Is first, why do we choose to put the fluorine on the sugar at this position? Because first, we know that the interaction between the proteins and the RNA happens between the residue and the base. So we don't want to disturb this interaction by putting the fluorine on the sugar rather than on the base. This is the first thing. And it has been shown in this paper that putting the fluorine in this position doesn't affect the thermostability of the RNA. Okay? The other thing now, why did we choose G6 and A20? Because if you do some literature about SRSF1, A1, and SAM68, you will know that those two proteins are more expected to bind AGG motifs, while SAM68 is known to bind multi-A motif. So we thought, okay, the most probable position binding sites are here, so let's label the G6 and the A20 and see. So we receive our RNAs, we do a fingerprint of the controls, then we add protein 1 with the RNA, protein 2 with the RNA individually, then we put pairs of protein with one RNA and see what happens to look for competitive or concurrent binding. You will see this in a minute. So these are our controls. We have doubly labeled RNA2 in G6 and A20, and to assign the two peaks, we ordered this RNA1 with only labeled in G6. And this is the only assignment you will see. And this is the advantage here, is that there is no assignment. Sorry for CCPN, but yeah, this is the advantage of the method. So we just overlay both RNAs, and you can see that there is a common peak here. And we know that this peak around minus 200 is for the G6, and the peak on the right here is for the A20. This is in buffer. Then we also wanted to work in nuclear extract. Why? Because we collaborate with Ian Epron Lab, who are working by single molecule spectroscopy. They work with big molecules, and they do splicing because the splicing occurs in the nucleus. So we need to add some nuclear extract. They add 40% of nuclear extract, and we want to compare our results. And this is why we need to work in the same conditions. So 30% of nuclear extract to work in physiological conditions. So even in a viscous sample, we still have the signal. It persists, and we are happy with that. So we decided to carry on and work in buffer and in nuclear extract. Okay. So we started adding one protein individually, and we can see here, these are our controls in buffer and nuclear extract. What we did is to calculate the integral of each peak here. We gave 1 or 100% to each peak. And then we measure the ratio of the integral of each peak after and before adding the protein, and monitor like that the decrease of the peak. Now, if you add SAM68 individually to the RNA, you will have 40% of decrease on this peak. Why? While the peak of G6 stays untouched. And we have similar results in buffer and nuclear extract. And this is what we are expecting. We are expecting SAM68 to bind this side and not this side. In the opposite, if you add SRSF1 or A1, this peak completely disappears. So you have 0%, 100% of decrease, while the other one is less affected or completely untouched. And this is also what we expected. We expected SRSF1 and A1 to affect the peak of the G and not the one of the A. So we're happy with that and carried on. Now adding both proteins, a pair of proteins, SAM68 and A1, for example. So these are the results with individual proteins, and these are the pairs of proteins added to one RNA. So same thing, controls in buffer and nuclear extract integrals. and then we add proteins, SUM68 and A1. And here you see something nice, which is a combination of the famous 40% decrease of SUM68 here, combine it to the 0% of A1. Same thing in buffer, you have this combined to this. What we don't see when we add only one protein, which is good. It means that both proteins are binding, so both proteins give the effects on the RNA. No competition between SUM68 and A1. However, if you add now SUM68 and SRSF1, same thing, buffer, nuclear extract. These are the individual proteins added, and these are the pairs of proteins. And you can see that here we start losing the effect of SRSF1, but it's very obvious in the nuclear extract that we lost the effect of SUM68, the famous 40% of decrease, and the 0% for which we have used the of SRSF1. So we lost these effects in the nuclear extract. And we want to confirm this using EMSA. EMSA is just a shift. It's where you put you monitor the shift of your RNA on a native gel without already proteins and see if you have formation of complexes. So you have the free RNA, and then you add your proteins in buffer or in nuclear extract. And in buffer, we see similar results. Let's see. Free RNA, then you add SUM68, you have binding, SRSF1, you have a complex, SUM68 and SRSF1, you have complex. This is what we see here. Then when you go to nuclear extract, this is the free RNA. If you add SUM68, you still have binding, SRSF1, you still have binding. If you add both proteins now, you lose completely the complex and you find something similar to the free RNA. So it's like amazing how EMSA reflects completely the results of the RNA. So results confirmed. We were happy to see both a situation where there is probably competition here. We don't know. We need to investigate more to understand this mechanism and concurrent binding in the previous case. But we were not very happy with this picture because once you go to the nuclear extract, the physiological conditions, even sometimes in the buffer, you still having those peaks appearing here, which are most probably degradation peaks, because if you do pseudo-2D, you monitor those peaks through time, you see that after 20 minutes, they start appearing and then they increase, while your signal decreases with time until everything disappears and you have this big, large peak here. So we know they have degradation peaks, and what we need to do is to work fast. We need like some experiments of less than 20 minutes without losing signal to noise. This is a big challenge for us. So we talked to our collaborator, chemist, Alex from Glenbury Lab, suggested to overcome this, to replace the fluorine with an OCF3 group. Why? For two reasons. First thing, we will get rid of the G coupling between the fluorine and the nearby protons here. So instead of having this multiplet, we have a nice peak in the middle, and then we increase the number of fluorine. So he made this phosphoramidite which is not available commercially, and sent it to the company that inserted it in the same position as the A20 in my RNA2. And we wanted to compare RNA3 to RNA2. And indeed, if you replace the fluorine in RNA2 with an OCF3 and RNA3, you have an increase of 70% of the signal. We have a nuclear extract in 30 minutes. It's 30 minutes, and that's why you still have a small degradation peaking here. So what we did, we went to 7 minutes nuclear extract, nice spectrum, and we wanted to see the interaction with SUM68 using the OCF3. And yes, if you add the protein, you still have this decrease of the A20 when you add the protein. And you can notice here that I'm using half of the concentration that I used before with the fluorine. So we were happy with this. The conclusion for this first part is that using the anamide fluorine, we were able to obtain an NMR signature for each protein. We saw a concurrent binding for SAM68 and A1, no binding in the nuclear extract, at least for SAM68 and SRSF1. Is it competition? Do the protein bind together instead of binding the RNA? We need more investigation for that. Then we know now that it's possible to investigate pairs of proteins binding to one RNA using the NMR of purine, and OCO3 RNAs can solve the degradation issues. Now, we were happy to use this oligomer to validate the technique, but as I mentioned before, we are collaborating with people working with single molecule spectroscopy, so they use big molecules, big RNAs of at least 120, 200, 500, 600. So the minimum is 120. So to be able to compare our results, we need to move to something larger, which is at least 120 nucleotides in this case. But now, when you use NMR and you want to move to something, to a large molecule, you have different challenges. The first thing is you have to be prepared to have this peak broadening or loss of signal. And the other thing is this is chemically synthesized. Are the chemists now able to still make as big, large molecule? Because we know that over 100 nucleotide, the chemical synthesis becomes tricky. So to confirm this, we just ordered this RNA of 120 bases with the same position as in the small oligomer of 21 nucleotides, the A20, but here it's A49 because the RNA is bigger. And we wanted to analyze it by NMR. They were able to make it, and they sent it to us, and we analyzed it. After 45 minutes at 50 micromolar, we managed to obtain a signal which was at the same shift as the A20 in the small oligomer of 21 men. So we're happy with this. It means we can carry on and use 19F to analyze the interaction between the RNA and proteins in the large 120 nucleotides. The problem is the yield and the cost, because we ordered 100 micromole, we received 3% of it, and this is the cost. Especially that what we want is to cover more binding sites of our proteins, so we want to put a fluorine each 10 nucleotides. And if you ask for the code for this, it's 24,000 pounds. So we needed an alternative to make this large multifluorinated RNAs. We asked again our chemists, collaborators, and they suggested to use splinted ligation. And in this case, it's double splinted ligation, using this protocol published here. What's splinted ligation? It is, instead of ordering this beast here, you split it to 3, the blue, the red, and the green here. And then you need a splint, which is an RNA, which the sequence has to be complementary to the end of RNA1, RNA2, and the beginning of RNA3. Then you anneal to bring, when you anneal, this splint will bring 3 portions together. Of course, you put whatever you want in RNA2. We need fluorine, so we put fluorine, but you can put OCF3, methoxy. You can also, if you want to segmental labeling, for example, you can label this with P13 and label it and ligate it, with 2 unlabeled portions. Anyway, so the splint will bring these 3 portions together when you anneal. Then you add a ligase, which is a bacteriophage ligase, that will join the edges together. Of course, you need those edges here, 5' to be phosphorylated to let the enzyme do the work. Then, once these are joined, you need to heat to remove your splint, and you end up with this product. You say, okay, let's try. And we try. Indeed, you obtain some single ligation products, which are RNA2 plus 3, RNA1 plus 2, but the major product was the 120, which comes from RNA1 plus 2 plus 3. Then, of course, you don't want this, and you don't want a splint in your sample, so you need to purify it using ion exchange chromatography. You isolate your sample. Then I made a large scale of this to obtain 24 micromolar and analyze this by NMR. And we had a nice peak. which the shift was exactly the same as the one that we bought, because they're different, because here we don't decouple and here we decouple. That's why it's sharp and nice. And said, okay, so we obtained a ligated fluorinated RNA, which with a nice peak, with a shift where we want. Let's see if it's functional, and let's add a protein, SAM68 and KKH again. It's just the binding domain that we've done. And we have this appearance of the peak. Knowing that this position is the same position A20, where we know that SAM68 is supposed to bind. Okay, we will have this. Now, now we need to make this by ligation. The challenge that we can face here is the overlapping signals, because you can make this list with all the fluorinated bases by ligation. It can take 1 year of work, and then you can cry in the corner because all the signal will overlap in one place, and you don't know which signal corresponds to which base. So before doing that, we split this to 9 portions, because it has 9 fluorines. Every portion has 1 fluorine, and we are trying to eliminate the overlapping one. So we had many nice signals. Many of them already don't overlap, which is good. The other challenge that we have is when you go from a small, for example, this is the blue one, is RNA1. When you split it to 3 portions, each portion will have a nice signal, almost, except this one. But then when you go to bigger size, of course, you have this broadening of your peaks. This is another challenge, which is not only due to the size, because we think, it's not a thing, we really saw it by experiment, that we have, it's due to the heterogeneity, of our sample, that where we have many conformers existing at the same time. Here, this is a published work that shows that for the same base, G here, for example, or U, either you are in a double strand or single strand, you will have different shifts and different conformers. So fluorine is very sensitive to structure, and you have to be careful with this when you order your fluorinated RNAs. We confirmed this just by heating. So if you take this portion that I showed before, and you heat it to 60 degrees, you obtain your 3 very nice peaks corresponding to your 3 bases. But obviously, you can't add proteins here because we are at 60 degrees, fortunately. So what we did, we slow cooled it. And if you slow cool, you have a very, much better signal than here anyway. And this is how, and here you can add your proteins because we are at 30 degrees. And this is how we are planning to overcome this problem of presence of conformers in the solution. Yeah, so this is the future work, is to eliminate the overlapping signals of the fluorinated bases and make a final multifluorinated RNA using splint ligation, then map the binding site of each protein. We already overexpressed 6 of them in the lab, and then look for any concurrent or competitive binding of this protein to our RNA. That's it. I want to thank people in my group, our collaborator in the Electron Lab, Andrew Hudson, Glenn, Burley. I want to thank Alex for the OC3 RNAs, John for the ligation, Alastair and Marina, Fred Muscat for NMR, and Sharon for cloning the plasmids, and Antoine Cléry and Frédéric Alain for SRSF1 clones. And you for your attention. This is our Splice Select network. If you're interested in our work, you can visit our site. Thank you for your attention.